Tonight, imagine a body that is not built to last, but built to leave traces behind. In humans, aging is not simply the clock running down. It is the bill that arrives when evolution stops paying attention. After the years when reproduction mattered most, repair becomes negotiable, and small failures are allowed to pile up like dust in a quiet room. Yet in the same world, some animals seem to live inside a different bargain. They keep their tissues steady, their functions calm, their decline strangely delayed. Not because nature loves them more, but because their lives make different demands, and their bodies can afford a different kind of maintenance. The cold thought is this: what looks like aging may be less about time, and more about what your species was worth at the only ages that counted. So, if some animals do not age like humans, what does that imply about what humans were designed to lose? And why was that loss acceptable in the first place? Aging is often described as if it were a single, universal clock, ticking at the same steady rhythm for every living being. Yet when biologists compare species across oceans, forests, and deserts, that image begins to soften. Some organisms show a clear pattern of gradual decline after maturity, with rising risk of disease, frailty, and death as the years pass. Others display a far subtler change. Their mortality risk does not rise sharply with age. Their physical functions remain comparatively stable across long stretches of time. This difference does not mean they escape death, nor that time forgets them. It means that aging, as measured by increasing vulnerability over time, does not unfold in the same way for all forms of life. For humans, the pattern is familiar. After reproductive maturity, the body slowly accumulates cellular damage. Repair systems continue to operate, yet they do so within limits. Proteins misfold more often, DNA errors persist, tissues regenerate more slowly. The arc is gentle at first, then more visible. By contrast, certain long-lived turtles, some species of rockfish, and large marine mammals exhibit what researchers call negligible senescence. Their probability of dying does not accelerate dramatically simply because they have reached an advanced age. The difference is statistical, not mythical. It is visible in population data gathered over decades, sometimes centuries. To understand why this matters, we must return to the beginning of the human story. Early members of our species lived in environments shaped by predators, infections, accidents, and scarcity. In such settings, few individuals reached what we would now call advanced old age. The average life expectancy at birth was low, not because human biology could not in principle sustain longer life, but because external hazards removed many lives before slow internal decline had time to dominate. When most individuals die from environmental causes, the evolutionary pressure to perfect long-term maintenance weakens. Genes that ensure survival through childhood and reproductive years are strongly favored. Genes that would provide benefits only after those years face a different calculus. If they offer no advantage during the reproductive window, natural selection has little opportunity to refine them. Aging, in this sense, is not a sudden failure but a gradual shift in biological priority. The body continues to function, yet it is not optimized for indefinite upkeep. In species where adults experience lower external mortality, the situation changes. If individuals routinely survive for many years after maturity, then traits that support long-term stability can influence reproductive success across a broader span of life. Over generations, this can shape a different aging curve. The contrast does not imply intention or design in a conscious sense. It reflects accumulated outcomes of survival and reproduction across deep time. When we look at humans today, living in societies where medicine reduces infection and injury, we are witnessing bodies shaped in a harsher past, now extending into ages that were once rare. The familiar human pattern of aging, with its steady rise in chronic disease, becomes more visible because external dangers have receded. Other animals, shaped by distinct ecological conditions, reveal alternative statistical patterns. Aging, then, is not a single law imposed evenly on all life. It is a spectrum of outcomes emerging from how long organisms typically survive, how they reproduce, and what their environments demand. If some animals do not age like humans, the difference begins not in mystery, but in the varied histories written into their bodies and into ours. When scientists speak about aging, they do not begin with wrinkles or grey hair. They begin with patterns in numbers. In many species, If we follow a large group from birth to death and measure the chance of dying at each age, we observe a steady rise after maturity. This rising curve is called senescence. It reflects increasing vulnerability to internal failure over time. In humans, the curve becomes especially visible in later adulthood, when chronic illnesses such as cardiovascular disease, neurodegeneration, and metabolic disorders grow more common. The body does not collapse all at once. Rather, the probability of breakdown gradually increases. This statistical pattern is so familiar that it is easy to assume it must be universal. Yet careful long-term studies reveal exceptions. Certain long-lived turtles, some deep-sea fish such as rockfish, and large marine mammals show mortality curves that remain relatively flat across adult life. Their risk of death does not accelerate sharply simply because they have grown older. This phenomenon, described as negligible senescence, does not mean they are immortal. They can still die from predation, disease, or environmental change. It means that aging, defined as a rising risk of internal failure with time, progresses differently. To understand why this difference exists, it is important to distinguish lifespan from aging rate. Lifespan is the maximum or average number of years a species can live. Aging rate refers to how quickly the risk of death increases as time passes. A species may have a moderate lifespan but a slow aging rate, or a long lifespan with noticeable decline late in life. Humans live longer than many mammals of similar size, yet our aging rate after middle adulthood is clearly measurable. The distinction matters because it shifts the question. We are not asking why some animals live longer in absolute years. We are asking why their bodies maintain stability longer before internal decline accelerates. When we return to the early human condition, the contrast gains clarity. For most of human evolutionary history, survival depended on mobility, cooperation, and rapid adaptation to changing landscapes. Hunter-gatherer groups faced injuries, infections, and nutritional fluctuations. Under such pressures, few individuals survived long enough for slow, late-life decline to shape reproductive outcomes significantly. Natural selection operated most strongly on traits expressed before and during the years of childbearing and child-rearing. In this context, modest imperfections in late-life repair systems would not have been heavily penalized. By contrast, in species that routinely survive environmental hazards well into advanced age, traits that stabilize tissues and delay internal deterioration can contribute to reproductive success across longer spans. For example, animals with slow growth, delayed maturity, and extended parental investment may benefit from durable bodies that remain functional for decades. Over evolutionary time, this shifts the balance of investment toward maintenance. The difference is subtle but cumulative. It does not produce eternal youth. It produces a flatter mortality curve. The human curve, shaped in a world where external risks were high, reflects a compromise. Our bodies allocate resources to growth, cognition, reproduction, and immune defense in early and midlife. Repair mechanisms remain active, yet they are not infinitely reinforced. As modern conditions reduce external threats, we see more clearly the internal limits of that allocation. The comparison with other animals does not reveal a hidden secret of immortality. It reveals variation in how life histories are written into flesh. Aging, measured as rising vulnerability, is one possible outcome among several. The human version of that outcome carries the imprint of environments where surviving long enough to reproduce mattered more than sustaining flawless function into extreme old age. If aging is defined as a rising risk of internal failure over time, then the next question is not why decline exists, but why it accelerates in some species and not in others. To approach this calmly, it helps to imagine the body as a system that constantly balances two demands. One demand is immediate performance, growth, movement, reproduction, and defense. The other is long-term maintenance, repairing damaged DNA, clearing misfolded proteins, replacing worn-out cells. Both require energy. No organism has unlimited reserves. In humans, the balance gradually shifts as adulthood progresses. Repair continues, but it is never perfect. Small imperfections accumulate, collagen fibers stiffen, stem cell pools narrow, immune responses become less precise. These changes are measurable at the cellular level long before they become visible in posture or memory. Over decades, the accumulation alters the probability of disease. The mortality curve rises. In species with negligible senescence, the balance appears to be distributed differently. Some turtles maintain stable reproductive output even in advanced age. Certain long-lived fish show little increase in mortality risk across many years of adulthood. The difference is not that their cells never experience damage. It is that the rate of repair, replacement, and stability remains proportionally aligned with the damage incurred under their ecological conditions. When environmental hazards are low and metabolic rates are slow, the pace of internal wear may also be slower. The statistical signature of aging becomes flatter. To see why humans follow a different trajectory, we must look again toward our origins. Early Homo sapiens evolved in environments that rewarded flexibility and social cooperation. A large brain required sustained energy input. Childhood was prolonged, and learning extended over many years. These traits offered advantages in complex landscapes, yet they also shaped how resources were allocated within the body. Energy invested in neural development and reproductive success during prime years could not be infinitely invested in cellular repair that would matter most decades later. The compromise was subtle. It did not prevent individuals from reaching older ages. Archaeological remains show that some early humans lived into what we would recognize as late adulthood, but they were fewer in number, and their survival depended heavily on social support within small groups. The population structure of those communities skewed young. Under such conditions, genetic variants that improved survival before and during reproductive years carried the greatest weight. Variants that might have extended flawless function far beyond that window exerted weaker influence. The result is not a flaw in human design. It is a reflection of historical probability. When most individuals did not regularly survive into advanced old age, the evolutionary return on investing heavily in extreme longevity was limited. Other species, shaped by different mortality patterns, faced different probabilities. Over many generations, these probabilities sculpted distinct aging curves. Aging, then, is not a moral failure of biology. It is a pattern emerging from trade-offs embedded in ecological history. Humans display one version of that pattern, written in tissues that were shaped for survival in unpredictable landscapes. Some animals display another version, shaped in environments where the rhythm of life unfolded more slowly and external threats were less abrupt. The contrast begins not with mystery, but with how long bodies were expected to endure under the conditions in which they first took form. In evolutionary biology, there is a quiet principle that helps explain why aging unfolds the way it does in humans. Natural selection does not aim for perfection across all ages. It acts most strongly on traits that influence survival and reproduction during the years when individuals are likely to leave offspring. Once that window narrows, the force of selection weakens. This principle is not philosophical. It is visible in population models and genetic studies. Genes that enhance early life survival or fertility spread efficiently through generations, even if those same genes carry subtle costs that appear later. If the negative effects emerge after most reproduction has already occurred, natural selection has limited opportunity to remove them. The body, therefore, is not engineered for indefinite upkeep. It is shaped for sufficient durability to reach and sustain reproductive contribution under typical environmental conditions. This idea forms the foundation of what is known as the disposable soma theory. The term soma refers to the body's non-reproductive cells, the tissues that carry out daily function. According to this framework, organisms allocate energy between reproduction and maintenance. Because energy is finite, increasing investment in one domain often reduces the other. In species where survival to older ages is common and reproduction occurs over extended periods, stronger investment in maintenance can evolve. In species where external mortality is high, the balance tilts toward earlier reproduction and adequate, but not maximal, long-term repair. When we examine early human environments, the relevance becomes clearer. Hunter-gatherer populations faced injuries, infections, and unpredictable climates. Anthropological data suggest that while some individuals reached older adulthood, many did not. Infant mortality was high. Accidental death was common. Under such circumstances, genes that improved early immune function, faster growth, or greater reproductive success would be strongly favored. Genes that offered benefits primarily at ages rarely reached would have less impact on reproductive outcomes. The result is a biological compromise. Human tissues possess sophisticated repair mechanisms, DNA proofreading enzymes, antioxidant systems, cellular cleanup pathways. Yet these systems are not infinite. Over decades, damage accumulates because maintenance is optimized for expected lifespan under ancestral conditions, not for the extended lifespans now possible in modern societies. By contrast, certain long-lived animals inhabit ecological niches with lower adult mortality. Large turtles protected by shells or whales with few natural predators experience different statistical pressures. When adults reliably survive for many years, traits that enhance durability into later life can influence reproductive success across a broader span. Selection can, over time, reinforce more robust maintenance systems relative to body size and metabolic rate. The divergence in aging patterns, then, reflects differences in how long adults typically survived in their environments. It is not that humans were meant to decline. It is that, historically, survival beyond certain ages was less predictable, and therefore less central to gene propagation. As modern medicine reduces external causes of death, we see more clearly the gradual internal trade-offs embedded in our biology. The question of why some animals do not age like humans begins to resolve into a question about which ages mattered most in each species' evolutionary past, and how strongly natural selection acted on maintaining the body beyond those ages. If natural selection acts most strongly on traits expressed before and during reproduction, then the timing of biological effects becomes central. Some genes carry benefits early in life but impose subtle costs later. This pattern is known as antagonistic pleiotropy. A single genetic variant may increase fertility, strengthen early immune response, or accelerate growth during youth, while also increasing vulnerability to disease decades later. Because early advantages directly influence reproductive success, such genes can spread widely, even if their late-life consequences are harmful. The logic is statistical rather than intentional. If most individuals in ancestral populations did not survive into very old age, then late-acting disadvantages would rarely affect how many offspring were produced. Over many generations, this dynamic shapes aging patterns. In humans, evidence for antagonistic pleiotropy appears in multiple systems. Hormones that promote rapid growth and sexual maturity in adolescence may also increase long-term metabolic strain. Robust inflammatory responses protect against infection in youth, but, when chronically activated, can contribute to cardiovascular disease and other age-related conditions. The immune system that once defended against pathogens in small, mobile groups now operates in environments with different exposures and longer lifespans. What once served immediate survival can, in extended old age, become a source of gradual decline. When we consider the early human condition, the relevance becomes more grounded. For most of our evolutionary history, reproduction occurred in early and middle adulthood. Survival beyond that period was variable. Grandparental support likely offered social advantages, yet the proportion of individuals reaching advanced ages remained limited compared to modern societies. Under such demographic structures, selection favored traits that ensured successful maturation, reproduction, and child-rearing within a relatively constrained time frame. Genetic variants that would have optimized tissue maintenance into the 8th or 9th decade would rarely influence overall reproductive output. In contrast, species that routinely experience lower adult mortality across extended spans encounter a different calculus. If individuals remain reproductively active for many years, then genetic factors influencing health in later adulthood can significantly affect total offspring production. In these contexts, alleles that improve long-term cellular repair or metabolic stability may be reinforced more consistently across generations. The difference is cumulative and subtle. It does not require conscious direction, only differential survival and reproduction over time. Human aging patterns, therefore, reflect the layered effects of genes that were advantageous in youth but less refined for advanced ages rarely reached. The accumulation of late-acting vulnerabilities is not evidence of biological negligence. It is evidence of historical probability. As environmental hazards decreased in recent centuries through sanitation, nutrition, and medicine, more individuals began to live long enough for late-life genetic trade-offs to manifest clearly. Conditions such as neurodegenerative disease and certain cancers become more visible not because they are new, but because more people survive into the age ranges where their risk rises. When comparing humans with animals that age more slowly, the contrast can be traced back to these differing demographic histories. The ages at which reproduction typically occurred, and the likelihood of surviving beyond them, shaped how strongly natural selection refined maintenance systems. The divergence in aging curves emerges from when life most often ended, and when it most often continued. Another way to understand the weakening force of natural selection in later life is to consider how probability changes with age. In any population exposed to predators, disease, and accidents, the number of individuals alive decreases steadily over time. If we imagine a cohort of early humans born into a Pleistocene landscape, many would not survive childhood. Others would fall to injury or infection in adolescence and early adulthood. By the time the group reached what we now call advanced old age, only a small fraction would remain. From the perspective of gene transmission, the influence of traits expressed at very late ages would therefore be diluted simply because fewer individuals lived long enough to express them. This demographic reality alters the intensity of selection across the lifespan. Traits that affect survival or fertility at age 20 can influence the genetic composition of the next generation profoundly. Traits that affect health at age 70, in a context where relatively few reach 70, would have a weaker evolutionary signal. Over thousands of generations, this difference accumulates. The body becomes finely tuned for the years when survival and reproduction most strongly shape lineage continuation, while later years receive less precise optimization. This does not imply that older individuals lacked importance in human groups. Anthropological studies of hunter-gatherer societies suggest that elders often contributed knowledge, childcare, and social stability. Grandparental support can enhance the survival of grandchildren, a concept sometimes described as the grandmother hypothesis. Yet even in such systems, the demographic distribution remained weighted toward younger ages compared with modern populations. The evolutionary pressure to perfect late-life cellular maintenance would still be limited by how many individuals typically survived into those years. When we compare this with species that show negligible senescence, the demographic contrast becomes significant. Large marine mammals, for example, often experience relatively low adult predation. If individuals routinely survive decades beyond reproductive maturity, then the proportion of the population living at advanced ages is higher. Under these conditions, genetic variants that enhance long-term tissue integrity can influence reproductive success, not only directly, but also through extended social roles or repeated breeding opportunities. The statistical weight of late-life performance becomes stronger. In humans, by contrast, extended longevity is a recent demographic shift. Improvements in hygiene, vaccination, and trauma care have altered survival curves dramatically within only a few centuries. Our bodies, however, remain shaped by much older distributions of risk. The increasing prevalence of age-related disease in modern societies is partly a reflection of this shift. More individuals now survive into decades where the evolutionary refinement of maintenance systems was historically less intense. The divergence between human aging and the flatter curves observed in some animals thus emerges from differing patterns of survival across age. The ages that were once rare have become common for us, revealing biological trade-offs that were long embedded but less frequently observed. Bodies do not age in isolation from the environments that shape them. Every species carries within its tissues a record of the risks it most frequently faced. Temperature, predation, food availability, and physical terrain all influence how survival unfolds across a lifetime. When external mortality is high, many individuals die from forces outside their own biology before slow internal decline becomes decisive. When external mortality is low, more individuals survive long enough for gradual cellular damage to matter. This ecological difference quietly alters how energy is allocated between immediate performance and long-term maintenance. Consider large tortoises that inhabit islands with few predators. Their heavy shells provide substantial protection. Their metabolic rates are slow. Food sources, though sometimes limited, are relatively stable compared with the fluctuating pressures faced by migratory mammals or small prey animals. In such contexts, adults often survive for many decades. Because they are likely to remain alive and reproductively active for extended periods, traits that preserve tissue function across long spans can contribute to lifetime reproductive output. Over evolutionary time, this pattern supports more durable maintenance systems relative to metabolic demand. The result is a mortality curve that rises slowly, sometimes so gradually that it appears nearly flat for long stretches. Humans emerged in very different settings. Early Homo sapiens occupied landscapes marked by predators, competition, climate shifts, and infectious disease. Mobility was essential. Energy had to be directed toward growth, cognition, immune defense, and reproduction under uncertain conditions. Injuries from hunting, childbirth, or conflict could be fatal without modern medical intervention. In such environments, many deaths occurred independently of internal aging processes. A broken bone, a severe infection, or prolonged famine could end a life well before gradual cellular deterioration became relevant. When external hazards dominate mortality, natural selection prioritizes traits that enhance early survival and reproductive success rather than perfecting maintenance systems that would only show benefits decades later. The contrast between these ecological contexts helps explain why some animals exhibit slower apparent aging. It is not that their cells are immune to entropy. It is that their life history unfolds in environments where surviving into advanced age is common enough to shape evolutionary investment. For humans, extended longevity became widespread only after the development of agriculture, sanitation, and medicine reduced many external risks. Our bodies, however, still reflect earlier conditions in which lifespan was constrained by hazards beyond cellular decline. As those hazards receded, the internal limits of our maintenance systems became more visible. The rise in chronic diseases associated with aging in modern societies partly reflects this shift from external to internal causes of death. When comparing humans with long-lived species such as certain whales or island tortoises, the key difference begins with ecology. The environment determines which ages are routinely reached and which are rare. Those probabilities, repeated over thousands of generations, influence how strongly natural selection refines the balance between reproduction and repair. Aging patterns, therefore, are not detached from landscape or climate. They are biological echoes of the environments in which survival once most often succeeded or failed. Metabolism offers another window into how environment shapes aging. The rate at which an organism converts food into usable energy influences how quickly cellular processes occur. Faster metabolism can support rapid growth, high activity, and early reproduction, but it may also generate greater oxidative stress within cells. Slower metabolism, often associated with colder environments or less active lifestyles, can reduce the pace at which molecular damage accumulates. This relationship is not absolute, yet broad comparisons across species suggest that metabolic tempo interacts with longevity in meaningful ways. Many long-lived animals that display negligible senescence share traits of relatively slow growth and steady, low-intensity energy expenditure. Their tissues are not constantly pushed to maximum performance. In contrast, species that must reproduce quickly in unpredictable environments often exhibit faster metabolic rates and shorter lifespans. Humans occupy an unusual position within this spectrum. Compared with other primates of similar size, humans live longer, yet our metabolic demands remain high because of our large brains and complex social behavior. The human brain consumes a significant portion of resting energy. Throughout evolutionary history, securing sufficient calories to sustain cognitive function required persistent foraging, cooperation, and adaptation. Energy directed toward neural maintenance was energy not available for unlimited cellular repair elsewhere. The trade-off is subtle but cumulative. In ecological settings where food availability fluctuated and physical exertion was routine, allocating resources to immediate survival and reproductive success would have carried priority. Over time, this allocation pattern becomes embedded in physiology. Certain marine mammals provide an instructive contrast. Large whales, protected by size and lacking many natural predators in adulthood, often have slower life histories. They mature later, reproduce over extended spans, and exhibit metabolic adaptations suited to stable oceanic conditions. Their survival probabilities at older ages are higher relative to many terrestrial mammals. Under such circumstances, maintaining tissue integrity for decades can enhance total reproductive output. The metabolic context supports long-term stability rather than rapid turnover. Returning to early human populations, the environmental variability of savannas and forests demanded flexibility. Seasonal changes affected food sources. Mobility required musculoskeletal resilience. Social complexity required cognitive investment. These pressures encouraged a balance between growth, brain function, and immune defense, calibrated to environments where external mortality remained significant. The internal cost of high metabolic performance may have been acceptable when many individuals did not routinely reach very advanced ages. In modern societies where caloric abundance and reduced physical threat are common, the same metabolic systems persist. Extended lifespans reveal long-term consequences of cumulative cellular stress that once remained less visible. The contrast with animals that age more slowly, therefore, reflects not only demographic differences but energetic ones. The pace of life encoded in metabolism shapes how damage accumulates and how vigorously it is repaired. Aging emerges from this interaction between energy use and environmental expectation. In species where energy expenditure is steady and external risk low, the aging curve can flatten. In humans, shaped by fluctuating conditions and high cognitive demands, the curve rises in a pattern consistent with our evolutionary tempo. Predation and protection form another quiet axis, along which aging patterns diverge. In ecosystems where adult animals face constant threat from predators, the likelihood of dying from external causes remains high across much of life. Under such conditions, the evolutionary advantage of investing heavily in cellular maintenance that would only benefit very late ages is reduced. If an organism is unlikely to survive long enough to experience slow internal decline, natural selection will more strongly reward traits that improve early escape, vigilance, reproduction, and rapid maturation. Small mammals that live under frequent predation pressure often follow this pattern. They reproduce early, produce multiple offspring, and exhibit relatively short lifespans. Their aging curves rise quickly, not because their biology is careless, but because the statistical expectation of long survival is low. By contrast, species that possess strong physical defenses or inhabit safer ecological niches encounter a different set of probabilities. A large tortoise protected by a thick shell faces fewer adult predators. A whale's size reduces vulnerability to most marine threats once it reaches maturity. When adult survival becomes more predictable, the evolutionary return on maintaining tissue integrity across many decades increases. Traits that support gradual, steady physiological stability can influence reproductive success over a longer span. The mortality curve in such species often remains flatter, reflecting the reduced impact of predation on adult death rates. For early humans, the ecological picture was complex. While adult humans possessed intelligence, tools, and cooperative strategies that offered protection, they were not insulated from danger. Predatory animals, intergroup conflict, accidents during hunting, and complications of childbirth contributed to mortality. Even if human adults were less vulnerable than smaller mammals, external threats remained significant enough that survival into advanced age was not guaranteed. Archaeological evidence indicates that some individuals did reach later adulthood, but population structures suggest that older age groups formed a smaller proportion of the community than they do today. In such demographic contexts, selection pressures favored robust early performance and social adaptability. The capacity to cooperate, share knowledge, and raise offspring effectively carried immediate evolutionary benefit, perfecting mechanisms that would maintain optimal tissue function far beyond the average lifespan of the time carried less weight. As environments changed through agriculture and technological development, the balance shifted. Improved shelter, controlled fire, and later medical interventions reduced many external causes of death. With fewer individuals lost to predation and infection, internal processes of aging became more prominent determinants of mortality. What appears today as intrinsic biological decline was once often overshadowed by external risk. Comparing humans with animals that age more slowly thus reveals how safety alters evolutionary calculus. When survival into older age is common, maintenance systems can be refined more extensively. When survival is uncertain, early life advantages dominate. Aging patterns are therefore ecological reflections, shaped by how protected adulthood typically was in each species' ancestral environment. Across the living world, species follow different rhythms of growth, reproduction, and survival. Biologists describe these patterns under the framework of life history theory. Rather than focusing on a single trait such as lifespan, Life history theory examines how organisms allocate time and energy across key stages: development, maturity, reproduction, and decline. Some species grow quickly, reproduce early, and produce many offspring in short intervals. Others mature slowly, reproduce later, and invest heavily in fewer offspring over extended periods. These contrasting strategies influence not only population dynamics, but also the pace and pattern of aging. Species that reproduce rapidly in unstable environments often experience what is called a fast life history. In such contexts, mortality from predation, climate variability, or resource scarcity can be high. The evolutionary response is to prioritize early reproduction before external forces intervene. Energy is directed toward rapid growth and fertility. Maintenance systems function adequately, yet they are not optimized for decades of survival because the likelihood of reaching very advanced age is low. The mortality curve rises steeply once internal deterioration begins to dominate. Aging appears rapid because life itself unfolds quickly. By contrast, species with slow life histories inhabit environments where adult survival is more reliable. They often mature later and produce fewer offspring, but invest more resources in each one. Because individuals are likely to survive longer, maintaining bodily integrity across many years becomes advantageous. Over generations, this can produce flatter mortality curves and slower observable aging. Long-lived seabirds, certain reptiles, and large mammals often exhibit aspects of this slower strategy. Their physiology reflects a pacing that aligns with extended reproductive opportunity. Humans occupy a distinctive position within this spectrum. Compared with many mammals of similar body size, humans have relatively slow development. Childhood is prolonged. Adolescence extends over many years. Reproduction does not begin immediately after physical maturity, but follows a period of social and cognitive preparation. At the same time, humans historically face substantial environmental risks that limited average lifespan. This combination produces a mixed life history profile. We are neither extremely fast nor fully slow in evolutionary terms. Our extended juvenile period reflects heavy investment in brain development and social learning, yet our ancestral environments imposed enough external mortality that late life optimization remains secondary to successful reproduction and child rearing. The human aging curve therefore reflects this intermediate strategy. We live longer than many species of comparable size, yet our risk of chronic disease increases noticeably in later adulthood. The body maintains functionality across decades, but eventually reveals trade-offs embedded in its developmental priorities. When compared with animals that show negligible senescence, the distinction becomes clearer. Those species often align more consistently with slow life histories under conditions of reliable adult survival. The pacing of their growth and reproduction allows maintenance systems to remain proportionally strong over long spans. Humans, shaped by fluctuating environments and complex social structures, display a different compromise. Aging, viewed through life history theory, emerges not as an isolated flaw, but as part of a broader strategy governing how time, energy, and reproduction were balanced across generations. To understand more fully how life history shapes aging, it is helpful to consider the timing of reproduction. In many species with fast life histories, reproduction begins soon after physical maturity. The window for producing offspring may be short, and the number of offspring high. Under these conditions, natural selection strongly favors traits that maximize fertility and early survival, even if those traits carry long-term physiological costs. The body is calibrated for rapid success rather than prolonged stability. Once reproduction declines, the evolutionary weight of maintaining perfect tissue function diminishes sharply. In slower life histories, reproduction often extends across a broader span of years. Individuals may produce fewer offspring at a time, but continue doing so over decades. When the reproductive window is long, traits that preserve bodily function into later adulthood contribute directly to lifetime reproductive output. Maintenance systems, therefore, can be refined more thoroughly because their benefits remain relevant for longer. This does not eliminate aging, but it alters its pace and visibility. The shape of the mortality curve flattens because adult survival and fertility remain linked over extended timeframes. Humans again present a layered pattern. Historically, reproduction began in early adulthood, Yet child-rearing required long-term parental investment. Offspring depended on adults not only for birth but for many years of care and teaching. This extended dependency created selection pressures favoring adults who could remain functional and socially engaged beyond initial reproduction. However, the reproductive window for women remained biologically limited, and overall adult survival was constrained by environmental risks. The result is a system in which some degree of post-reproductive survival carries social value, yet the intensity of selection on late-life maintenance remains moderate compared with species that reproduce across most of their lifespan. In species such as certain whales, reproduction can continue for decades, and adult survival into advanced ages is common. Under such circumstances, maintaining cardiovascular integrity, immune competence, and musculoskeletal strength directly supports continued reproductive contribution. Over many generations, this can reinforce physiological traits associated with slower senescence. The ecological context supports long adult phases in which reproduction and survival remain intertwined. Returning to early human populations, the balance becomes clearer. While older individuals likely contributed knowledge and social stability, the demographic proportion of individuals living well beyond reproductive years was limited. Selection could favor traits supporting effective parenting and social cohesion, yet the evolutionary incentive to perfect cellular repair systems into extreme old age was less pronounced. Modern increases in lifespan have revealed how these historical balances manifest today. As more individuals survive decades beyond reproductive cessation, age-related conditions become more common. The underlying life history framework remains visible. Our biology was shaped for extended development and meaningful adult contribution, but not necessarily for indefinite preservation. Aging patterns, when viewed through reproductive timing, reflect how long reproduction and survival overlapped in ancestral environments. Life history theory also invites us to consider the number of offspring produced and the investment placed in each one. Species that produce many offspring at once often cannot afford to allocate extensive resources to long-term maintenance of the adult body. Energy must be divided among growth, reproduction, and immediate survival. In environments where offspring mortality is high, producing more young increases the probability that some will survive. Under such pressures, the adult organism may function as a vehicle for rapid gene transmission rather than as a structure designed for prolonged durability. Aging in these species often appears earlier and progresses more quickly because evolutionary success depends less on extended adult lifespan and more on early reproductive output. In contrast, species that produce fewer offspring and invest heavily in each one follow a different pattern. When the survival of offspring depends on sustained parental care, adults must remain functional for longer periods. Physical stability, cognitive competence, and social integration become essential not only for reproduction but for ensuring that offspring reach maturity. Over generations, this can strengthen selection for traits that preserve adult health across extended spans. The aging curve in such species often rises more slowly, reflecting this prolonged overlap between survival and reproductive contribution. Humans again occupy a distinctive position. Compared with many mammals, human reproductive rates are relatively low. Gestation is long, infancy is prolonged, and childhood extends over many years. Each child requires substantial energy, protection, and social learning. This pattern aligns with aspects of a slow life history. Adults who remain capable of providing care and knowledge confer advantages to their offspring. Archaeological and ethnographic evidence suggests that cooperative care within kin networks improved survival in early human groups. Grandparents, particularly grandmothers, may have contributed food, guidance, and support that enhanced the reproductive success of younger generations. Yet this social advantage operated within demographic constraints. While some individuals survived into later adulthood, the proportion of the population reaching very advanced ages remained limited. Environmental hazards continued to shape survival probabilities. The selective pressure to maintain perfect cellular repair into extreme old age therefore remained moderate rather than overwhelming. The human body reflects this balance. We possess robust immune systems, efficient wound healing in youth, and sophisticated cognitive capacities that support long-term social bonds. However, the gradual accumulation of molecular damage becomes more visible as decades pass, particularly in modern contexts where external mortality is reduced. The investment per offspring encouraged prolonged adult functionality, but not necessarily indefinite biological preservation. When comparing humans with animals that exhibit negligible senescence, the contrast highlights differences in reproductive strategy and ecological safety. In species where adults reliably survive and continue reproducing over long spans, investment in maintenance may extend further. In humans, shaped by variable environments and finite reproductive windows, aging unfolds as part of a compromise between parental investment, survival risk, and energetic limits. The pattern we observe today is an expression of how many offspring were produced, how long they depended on care, and how frequently adults survived long enough for late-life refinement to influence evolutionary outcomes. Beneath the visible signs of aging lies a quieter process unfolding within cells. Every day, in every tissue, damage occurs. DNA strands break and are repaired. Proteins fold and occasionally misfold. Mitochondria generate energy, and in doing so, produce byproducts that can alter nearby molecules. The persistence of life depends on the balance between injury and repair. In youth, repair mechanisms operate with remarkable efficiency. Cells divide, damaged components are removed, and tissues maintain structure with minimal visible decline. Over time, however, the balance shifts. Repair continues, but it becomes less precise. Small imperfections accumulate. In humans, this gradual accumulation underlies much of what we recognize as aging. One of the central elements in this process is the capacity for regeneration. Some tissues, such as skin and blood, renew continuously. Others, including certain neurons and cardiac cells, have limited regenerative potential. As stem cell reserves narrow and signaling pathways change with age, the ability to replace damaged cells slows. The body compensates for many years, yet the margin for error gradually decreases. Chronic inflammation, subtle immune dysregulation, and metabolic shifts begin to interact, increasing vulnerability to disease. When comparing humans to animals that age more slowly, differences in regenerative capacity become significant. Certain amphibians, such as axolotls, can regenerate limbs, portions of their spinal cord, and even parts of their heart. While they are not immune to aging, their capacity for tissue renewal is striking. Some fish species maintain high regenerative activity across long spans of life. These traits suggest that in certain evolutionary contexts, sustained regenerative potential offered advantages that were preserved and refined. The ecological background again matters. Species with lower external mortality and slower life histories may benefit from maintaining robust repair systems because individuals are likely to survive long enough to use them. In early human environments, however, severe injury often proved fatal regardless of regenerative capacity. A deep wound without modern sanitation could lead to infection. A fractured bone might limit mobility in ways that compromise survival. Under such circumstances, the evolutionary payoff of perfect limb regeneration would be constrained by environmental hazards. Instead, selection favored efficient wound healing, sufficient to support short to medium-term survival and reproduction. The human immune and repair systems are therefore powerful but calibrated within energetic limits. The maintenance of a large brain, complex social interaction, and sustained physical activity required substantial metabolic investment. Allocating additional energy toward indefinite regenerative potential might have imposed costs elsewhere. Over generations, the resulting compromise produced a system capable of decades of function, but not continuous renewal. In modern contexts, where medical intervention can prevent death from injuries that once proved fatal, the limitations of our regenerative systems become more visible. Chronic diseases associated with tissue degeneration emerge as leading causes of mortality. The contrast with animals that retain stronger regenerative capacity illustrates how different ecological expectations shape cellular investment. Aging, at the microscopic level, reflects not only the inevitability of molecular damage, but the evolutionary decision about how much energy to devote to repairing it over time. Within each human cell, specialized systems work continuously to preserve stability. DNA repair enzymes scan for breaks and mismatches. Proteasomes identify damaged proteins and dismantle them. Autophagy pathways recycle worn cellular components, allowing raw materials to be reused. In youth, These processes operate with coordinated precision. Cells respond swiftly to stress, and damaged elements are removed before they accumulate. Over time, however, the efficiency of these systems gradually declines. The reasons are complex and involve shifts in gene expression, epigenetic patterns, and cellular signaling networks. The result is not a sudden collapse, but a subtle imbalance between damage and restoration. As imperfectly repaired molecules accumulate, tissues begin to lose resilience. When researchers examine species that exhibit slower aging, they often find differences in these same maintenance pathways. Some long-lived animals show sustained activity of DNA repair enzymes well into advanced age. Others maintain more stable proteostasis, the balance of protein synthesis, folding, and degradation. These cellular characteristics correlate with flatter mortality curves observed at the population level. The connection between microscopic repair and macroscopic aging becomes clearer when viewed across decades. The persistence of cellular stability delays the onset of widespread tissue dysfunction. In early human environments, the evolutionary calculus surrounding cellular repair was shaped by expected lifespan and external risk. Maintaining high levels of repair requires energy and regulatory complexity. In settings where individuals frequently faced lethal injury or infection before advanced age, extreme investment in long-term molecular perfection may have offered diminishing returns. Instead, repair systems evolved to function robustly during the years most relevant to reproduction and child-rearing. They remain active throughout life, but their gradual decline reflects a balance between energetic cost and expected benefit. The human body also faces constraints linked to cell division. Each time a cell divides its telomeres, protective sequences at chromosome ends shorten slightly. Telomere shortening acts as a protective mechanism against uncontrolled cell growth, reducing cancer risk. Yet it also limits the replicative capacity of certain tissues over time. In species with different life histories, telomere dynamics vary. Some animals maintain telomerase activity in ways that support longer-term tissue renewal, though often with associated trade-offs. The balance between cancer prevention and regenerative capacity illustrates how repair systems must navigate competing risks. For humans, shaped by fluctuating environments and significant early life mortality, the selected balance prioritizes protection against immediate threats while allowing gradual late life decline. As modern conditions extend lifespan, these underlying balances become more visible. Age-related diseases reflect cumulative molecular changes that once had limited opportunity to influence reproductive success. The comparison with animals that age more slowly thus highlights differences in how long cellular maintenance remained under strong evolutionary refinement. Repair mechanisms in humans are sophisticated, yet calibrated to historical expectations of survival. Aging emerges from the gradual relaxation of precision in these systems as decades pass. Another dimension of regeneration involves not only the repair of molecules, but the renewal of entire tissues. In humans, certain organs retain modest regenerative capacity throughout life. The liver can regrow after partial removal. The skin repairs after injury. Blood cells are continuously replenished from stem cell reservoirs in bone marrow. Yet even these systems change with age. Stem cells divide less frequently. Their internal signaling becomes more variable. The surrounding cellular environment, known as the niche, alters in ways that reduce efficiency. Gradually, renewal slows. The body maintains continuity, but with increasing fragility. In species that display slower aging, patterns of tissue renewal can differ in subtle but meaningful ways. Some fish and amphibians preserve stem cell activity at levels that remain relatively stable across long spans. Certain reptiles show sustained growth or maintenance without the pronounced decline seen in many mammals. These differences are not absolute, nor do they imply immunity from deterioration. They indicate that the tempo of decline is shaped by how long regenerative systems remain under evolutionary pressure to perform. The ecological context again provides explanation. When adult survival is common and reproduction extends across many years, individuals benefit from tissues that remain robust over time. A whale that continues breeding into advanced age gains from cardiovascular and immune systems that remain stable for decades. A turtle that matures slowly and reproduces intermittently over long spans benefits from shells and organs that resist cumulative breakdown. In such environments, maintaining stem cell populations and repair mechanisms into later life influences reproductive success more consistently. For early humans, however, the landscape was less predictable. While adults could live into later years, many faced injuries, infectious disease, and nutritional stress that limited survival. Severe trauma often led to death regardless of regenerative capacity. Under these circumstances, the evolutionary payoff of perfect long-term tissue renewal was constrained by environmental risk. Selection favored repair systems sufficient to sustain survival through reproductive and caregiving years, but not necessarily optimized for many decades beyond. Modern medicine has altered this equation. Surgical techniques, antibiotics, and chronic disease management now allow individuals to survive conditions that once proved fatal. As a result, the gradual limitations of stem cell renewal and tissue repair become more visible. Degenerative conditions such as osteoarthritis, heart failure, and neurodegeneration reflect cumulative changes in tissues that were not historically exposed to such extended survival times across large populations. The comparison with animals that age more slowly underscores how regeneration is tied to expected lifespan in ancestral environments. Where survival into advanced age was common, sustained renewal carried evolutionary weight. Where survival was uncertain, repair systems were calibrated for nearer horizons. Aging, at the level of tissue renewal, reveals this historical calibration. The difference between humans and some longer-lived animals is not rooted in a single mechanism, but in how long regenerative capacity remains central to reproductive opportunity across generations. Among the many traits that distinguish humans from most other animals, the size and metabolic demand of the brain stand out. The human brain consumes a substantial portion of the body's resting energy, far more than in most mammals of comparable size. This energetic requirement begins early in life. Childhood is extended, and brain development continues for many years. Neural circuits are refined through learning, memory formation, and social interaction. The benefits are profound. Complex language, cooperation, planning, and cultural transmission. Yet this cognitive expansion carries biological cost. Energy devoted to maintaining neural tissue cannot simultaneously be invested elsewhere without limit. Every calorie used to sustain synaptic activity and neural repair represents energy that must be gathered, processed, and distributed within the constraints of a finite metabolism. In evolutionary terms, this allocation reflects priority. For early humans, enhanced cognition improved survival in unpredictable environments. Tool use, coordinated hunting, and social alliances increased the likelihood of reaching reproductive maturity and supporting offspring. Natural selection therefore strongly favored traits that supported brain growth and function during childhood and early adulthood. The energetic investment was worthwhile because it directly influenced reproductive success. However, this same investment shaped how other maintenance systems evolved. When energy is finite, trade-offs are unavoidable. The body must balance immune defense, muscle function, reproductive capacity, and long-term cellular repair. In species with smaller brains and slower metabolic rates, the energetic landscape differs. Certain long-lived reptiles and marine mammals allocate energy in ways that emphasize sustained tissue stability over decades. Their cognitive demands, while significant within their ecological niches, do not require the same proportion of metabolic expenditure relative to body size. This difference does not imply superiority or inferiority. It reflects distinct evolutionary priorities. For humans, the brain became central to survival. Extended learning periods allowed cultural knowledge to accumulate across generations. Yet the metabolic cost of this expansion likely influenced how much energy could be consistently directed toward indefinite tissue repair. Over decades, the cumulative impact of these energetic decisions becomes visible. Age-related conditions affecting cardiovascular, metabolic, and musculoskeletal systems may partly reflect the balance struck in deep time between cognitive advantage and systemic maintenance. When comparing humans to animals that age more slowly, the contrast often aligns with differences in metabolic tempo and organ investment. A large-brained primate with high social complexity faces distinct energetic demands compared with a slow-growing reptile protected by a shell. Early human populations, living in variable climates and competing for resources, benefited greatly from intelligence and cooperation. The selective pressure to sustain peak cognitive function through reproductive and caregiving years was intense. The selective pressure to optimize cellular repair into extreme old age was more moderate. Aging patterns, viewed through the lens of brain evolution, reveal how the extraordinary expansion of one organ reshaped the distribution of energy throughout the body. The energetic cost of a large brain does not operate only at rest. It shapes how the entire body responds to scarcity, stress, and aging. The human brain depends almost continuously on glucose and oxygen. Unlike some organs that can reduce activity under limited supply, neural tissue is highly sensitive to interruption. Throughout evolutionary history, this vulnerability required reliable energy acquisition. For early humans, securing sufficient calories meant sustained physical effort, cooperation, and adaptability. When food was scarce, the body entered conservation modes that protected brain function as a priority. Hormonal systems adjusted metabolism, sometimes at the expense of muscle mass or long-term tissue repair. This hierarchy of allocation reflects evolutionary logic, preserving cognitive capacity, improved chances of finding food, coordinating group activity, and avoiding danger. Over time, repeated cycles of energy, stress, and recovery may have influenced how maintenance systems were calibrated. If short-term survival and cognitive resilience were paramount, resources devoted to indefinite repair of peripheral tissues could remain limited. In species with slower metabolisms and smaller brains relative to body size, the energetic distribution differs. Certain long-lived animals can tolerate extended periods of low metabolic activity. Some marine mammals adjust oxygen use during deep dives. Certain reptiles reduce metabolic rate in colder temperatures. These adaptations lessen cumulative cellular stress over decades. When energy throughput is steadier and less concentrated in a single organ, maintenance systems may experience different selective pressures. The pace of molecular turnover and oxidative stress can be slower, contributing to flatter aging curves. Returning to early human societies, energy balance was rarely constant. Seasonal fluctuations affected food availability. Migration demanded physical exertion. Illness could reduce intake at critical times. In such environments, the body's priority was to sustain neural and reproductive function through variable conditions. Mechanisms that stabilized blood glucose and supported brain metabolism were strongly favored. The long-term cost of repeated metabolic strain on cardiovascular or endocrine systems would have been less visible if many individuals did not survive into advanced age. Modern conditions have altered this pattern. In many societies, caloric abundance replaces scarcity, yet the metabolic systems shaped for fluctuating energy remain active. Persistent high intake combined with reduced physical exertion can amplify stress on tissues over decades. Chronic conditions associated with aging, such as type 2 diabetes and cardiovascular disease, partly reflect this mismatch between ancestral metabolic calibration and contemporary environments. The contrast with animals that age more slowly underscores how metabolic architecture interacts with ecological expectation. A species whose survival rarely depends on sustaining an energetically expensive brain under variable conditions may allocate energy differently across tissues. In humans, the dominance of neural demand influenced the long-term distribution of repair and maintenance resources. Aging, therefore, can be seen as an outcome of how energy was historically prioritized, especially in service of cognition and social complexity. Beyond raw energy consumption, the structure of the human brain introduced another long-term biological cost: prolonged developmental vulnerability. Human infants are born neurologically unfinished. Compared with many mammals, they require years of care before achieving independence. This extended development reflects the complexity of neural wiring necessary for language, planning, and social awareness. During this time, a substantial portion of parental and communal resources is directed toward protecting and nourishing growing brains. Selection therefore favored adults who could sustain cognitive and social performance through the years when offspring remained dependent. The maintenance of mental clarity, coordination, and cooperative behavior in early and middle adulthood carried clear evolutionary value. However, this focus on supporting extended development also shaped broader physiological priorities. In environments where survival depended on social cohesion and knowledge transmission, sustaining functional cognition during reproductive and caregiving years was critical. Mechanisms that preserved neuroplasticity and immune competence during these phases were reinforced. Yet the intensity of selection declined as individuals moved beyond typical reproductive windows. Once offspring reached maturity, the direct reproductive advantage of further cognitive maintenance diminished. Late-life neurodegenerative processes, while deeply consequential in modern societies, would have exerted weaker evolutionary pressure in contexts where fewer individuals survived to extreme ages. In species with different developmental patterns, the allocation of resources can follow alternative paths. Animals that mature quickly and reach independence soon after birth do not require extended parental cognitive investment. Their neural development may be less prolonged, and the energetic burden of supporting a large plastic brain is reduced. In such cases, energy that might have been channeled into sustaining prolonged cognitive growth in humans can be distributed differently, potentially influencing long-term tissue maintenance. This does not mean that smaller-brained animals automatically age more slowly. Many do not. Rather, it highlights that the human life course, with its unusually extended childhood, required a specific configuration of energy and repair priorities. Archaeological findings support the idea that knowledge transmission and tool refinement accumulated gradually over generations. Elders who retained cognitive capacity could contribute to group survival by sharing experience about migration routes, seasonal cycles, and social alliances. Such contributions may have extended the functional importance of certain adults beyond reproductive years. Yet the demographic structure of early populations still limited the proportion reaching advanced old age. The balance remained weighted toward maintaining performance through the most reproductively and socially active decades. As medical advances increased survival into later life, age-related cognitive decline became more visible across populations. Conditions such as dementia reflect cumulative neural vulnerability that was historically less likely to manifest widely. The contrast with some longer-lived animals, whose ecological niches require less prolonged developmental investment, underscores how the human brain reshaped the aging landscape. Our extended childhood and high cognitive demand offered profound adaptive advantages, yet they also influenced how long-term maintenance was distributed across tissues and across the lifespan. When discussing animals that appear not to age like humans, it becomes important to clarify what not aging truly means. In scientific terms, negligible senescence refers to a pattern in which mortality risk does not rise steeply with chronological age. It does not imply biological immortality. Even species with flat mortality curves eventually die from disease, predation, environmental change, or random cellular events. The distinction lies in how strongly age itself predicts increasing vulnerability. In humans, the probability of dying from internal causes rises significantly after midlife. Chronic conditions accumulate, and physiological resilience gradually declines. Age becomes a powerful statistical factor. In certain long-lived species, by contrast, chronological age exerts a weaker influence on mortality probability across much of adult life. This contrast can be illustrated by comparing two types of risk. Extrinsic risk arises from external factors such as predators, accidents, or environmental hazards. Intrinsic risk stems from internal biological deterioration, including organ failure or progressive disease. In many early human environments, extrinsic risk dominated. Injury, infection, and conflict frequently ended lives before intrinsic aging processes became the primary determinant of death. As sanitation and medical care improved in modern societies, extrinsic risk decreased, allowing intrinsic processes to emerge as the leading causes of mortality. The human aging curve, therefore, reflects a transition from external to internal determinants of survival. In species with negligible senescence, extrinsic and intrinsic risks interact differently. Large tortoises protected by shells experience relatively low adult predation. If they avoid environmental catastrophes, their survival probability remains comparatively stable across decades. Internal deterioration progresses slowly enough that it does not sharply elevate mortality at specific chronological thresholds. The mortality curve remains flatter because intrinsic risk increases gradually rather than steeply. To understand why humans display a different pattern, it helps to return to demographic structure. In ancestral populations, the average number of individuals reaching advanced age was limited. Even if intrinsic aging processes were present, they often remained overshadowed by extrinsic threats. Selection pressures therefore operated within environments where the age-dependent rise in mortality carried less statistical weight. The body evolved to function effectively through the years most likely to be lived, not necessarily to eliminate intrinsic decline far beyond them. Modern human longevity has altered visibility but not underlying biology. As fewer individuals die from infection or trauma in early adulthood, more survive into decades where intrinsic aging processes manifest clearly. Cardiovascular disease, neurodegeneration, and cancer become more common not because they are novel, but because more people reach ages at which cumulative damage influences survival probability. The comparison with animals that age more slowly reveals how differences in ecological protection and demographic distribution shape the relative importance of intrinsic risk. Aging, understood statistically, reflects how strongly age itself predicts increasing mortality. In humans, age exerts a pronounced influence. In certain other species, the influence is gentler across much of life, illustrating alternative evolutionary calibrations rather than an absence of biological limits. If age does not predict mortality in the same way for all species, then the shape of survival curves becomes central to understanding the difference. In humans, demographic data from modern populations show a clear pattern. After early adulthood, the risk of death rises in a roughly exponential manner. This pattern, described mathematically in models such as the Gompertz law of mortality, reflects the accelerating influence of internal biological decline. The longer a human lives, the greater the statistical likelihood that accumulated cellular damage, systemic inflammation, or organ dysfunction will reach a critical threshold. Age becomes a powerful predictor of vulnerability. In species exhibiting negligible senescence, survival curves look different. Mortality risk may remain relatively constant across adult years, without the steep upward acceleration seen in humans. This does not mean individuals avoid decline entirely. It means that the rate at which intrinsic deterioration translates into death remains comparatively steady. For some turtles and certain fish species, studies suggest that older adults are not dramatically more likely to die in a given year than younger adults of the same species. The curve flattens rather than climbs sharply. The ecological implications of these shapes are profound. In early human societies, extrinsic hazards often shape survival curves more strongly than intrinsic aging. A hunter injured during a fall, a mother experiencing childbirth complications, or an individual exposed to infectious disease might die independent of slow molecular decline. As long as external mortality dominated, the steep intrinsic curve that becomes visible in modern data would have been less apparent in daily life. The demographic profile of such populations skewed toward youth and early adulthood. Selection pressures therefore responded primarily to survival probabilities within that earlier span. In animals with flatter intrinsic curves, the ecological environment often supports longer adult survival. A large marine mammal, once reaching maturity, may face few predators. Environmental conditions in deep ocean habitats can remain relatively stable compared to terrestrial climates. When adults routinely survive for decades, the proportion of the population reaching advanced age increases. Under these circumstances, traits that maintain physiological stability over long periods exert measurable effects on reproductive success. The statistical weight of older individuals grows, reinforcing maintenance systems across generations. For humans, modern public health has reshaped survival curves within a short historical window. Vaccination, antibiotics, sanitation, and improved nutrition have dramatically reduced early life mortality and many extrinsic causes of death. As a result, intrinsic aging patterns now dominate survival statistics. The exponential rise in mortality with age becomes clearly visible because more individuals survive long enough for intrinsic deterioration to manifest. The comparison with animals that age more slowly reveals how demographic history shapes the interpretation of aging. Where intrinsic risk remains relatively flat, aging appears less pronounced. Where intrinsic risk accelerates steeply, aging becomes central to the life course. These curves are not accidents. They are outcomes of ecological expectation and evolutionary calibration. It is tempting, when hearing that some animals show negligible senescence, to imagine a biological escape from time itself. Yet no known vertebrate avoids mortality indefinitely. Even species with relatively flat mortality curves eventually experience limits imposed by disease, environmental change, or random molecular events. The difference lies not in immortality, but in how gradually intrinsic risk accumulates. For humans, the steady rise in mortality with age creates a clear narrative of decline. We observe increasing frailty, higher incidence of chronic disease, and measurable loss of physiological reserve. The story appears linear because statistical probability intensifies in later decades. In animals with slower aging patterns, the narrative is less dramatic. An older turtle may resemble a younger adult in many functional measures. A long-lived fish may not exhibit sharply increased mortality simply because of chronological age. However, these individuals remain vulnerable to sudden environmental shifts, predation, or internal failure. Their flatter curves mean that aging contributes less strongly to annual risk, not that it disappears. Understanding this nuance is essential for avoiding misunderstanding. When humans compare themselves to such species, the contrast often feels stark because our intrinsic risk curve rises visibly in modern populations. Yet this visibility is partly a consequence of historical change. As external causes of death have diminished, intrinsic aging has become the dominant driver of mortality. In earlier human environments, the distinction between extrinsic and intrinsic causes was less clear in daily life. Many individuals did not survive long enough for age-related decline to dominate their experience. The gradual internal processes that now define late adulthood were once statistically overshadowed by injury, infection, and environmental stress. In species where adult survival has long been common, intrinsic and extrinsic risks have interacted differently across evolutionary time. When older individuals consistently form a meaningful proportion of the population, traits that reduce intrinsic deterioration carry measurable reproductive consequences. Maintenance systems may therefore be refined in ways that produce flatter mortality curves. This does not eliminate trade-offs, it redistributes them. Energy invested in prolonged stability may limit other capacities, such as rapid growth or high reproductive output. The diversity of aging patterns across species reflects these varied allocations. For humans, the steep rise in intrinsic risk with age reveals the historical balance struck between early life performance and long-term maintenance. As medical and social conditions extended lifespan, the underlying curve became more apparent. The comparison with animals that age more slowly underscores how survival patterns across generations shape the prominence of intrinsic decline. Aging, in this statistical sense, is not a universal script written identically in all bodies. It is a pattern whose visibility depends on how often individuals lived long enough for age itself to become the principal determinant of mortality. Long before written records, human bones quietly recorded the marks of aging. Archaeology offers a different lens on the question of why humans age as they do. Skeletal remains from prehistoric sites reveal patterns of wear, injury, and survival that help reconstruct demographic structure. Teeth worn flat from coarse diets, vertebrae showing signs of compression, joints bearing traces of arthritis, all testify that some individuals reached what we would now call later adulthood. Yet when researchers examine burial populations, they often find that relatively few individuals lived into advanced old age compared with modern societies. Infant mortality was high. Many adults died in early or middle adulthood. The age distribution of ancient communities was weighted toward youth. This demographic structure has implications for how aging influenced evolutionary outcomes. If only a small proportion of individuals survived beyond the reproductive years, then the selective pressure acting on late-life traits would have been limited. The archaeological record does not show an absence of older individuals, rather it shows that they were less common. In some sites, evidence suggests that older adults were cared for despite injury or disability. Healed fractures and survival with missing teeth imply social support within groups. Such findings indicate that human societies valued older members and that cooperation extended beyond immediate reproductive roles. However, the relative scarcity of advanced age within populations remains visible in demographic reconstructions. In contrast, species inhabiting safer ecological niches may consistently sustain higher proportions of adults reaching advanced ages. In those contexts, older individuals contribute directly to reproductive output, or social stability over longer spans. The proportion of the population exposed to late-life intrinsic decline becomes larger. Over many generations, this difference can influence how strongly maintenance systems are refined. Returning to early human environments, survival depended heavily on mobility, foraging skill, and resistance to infection. Severe injury could end a life despite social care. Climatic shifts and food scarcity imposed additional pressures. Under these circumstances, the probability of surviving into decades where chronic degenerative conditions would dominate was comparatively low. The aging processes that are so prominent in modern societies existed then as well, but fewer individuals lived long enough for them to define the population's mortality profile. The archaeological evidence, therefore, supports the idea that human aging patterns reflect historical probabilities rather than sudden modern decline. Our bodies evolved in contexts where extended old age was possible but not typical. As technological and medical advances increased the proportion of individuals reaching later decades, the biological trade-offs embedded in our physiology became more visible. Comparing skeletal records with the survival patterns of long-lived animals highlights how demographic weight shapes evolutionary pressure. Where older age is common across generations, late-life maintenance can influence lineage more strongly. Where it is rare, its influence diminishes. The bones of our ancestors remind us that aging in humans has always been present, yet its demographic prominence has shifted over time. Beyond the age distribution visible in skeletal remains, archaeology also reveals patterns of disease and degeneration that inform our understanding of aging in early humans. Bones sometimes carry signs of chronic inflammation, joint deterioration, or repetitive strain. These traces suggest that even in ancient populations, the processes we associate with aging were present. However, the frequency and context of these findings matter. When a small proportion of skeletons display advanced degenerative change, it implies that relatively few individuals survived long enough for such conditions to become widespread within the community. The biological mechanisms of aging were active, but their demographic footprint was limited by survival patterns. Dental wear provides another example. In many prehistoric populations, teeth were worn down significantly by abrasive diets. Some individuals lived long enough for enamel to erode deeply, yet complete tooth loss was less common than might be expected if large portions of the population regularly reached extreme old age. Similarly, evidence of advanced osteoporosis or extensive degenerative spinal disease appears, but not at frequencies comparable to those seen in modern aging societies. These observations align with the idea that while aging processes were biologically present, they influenced a smaller segment of the population overall. Environmental hazards often curtailed life before late-stage degeneration could accumulate widely. Social structures also shaped survival. Cooperative care, food sharing, and communal defense may have extended the lives of injured or aging individuals beyond what solitary survival would permit. The presence of healed fractures and survival with physical impairment indicates that early human groups supported vulnerable members. This support likely enhanced group resilience and knowledge retention. Yet such care operated within ecological constraints. Severe infections, untreated injuries, and climatic extremes still imposed limits on how long most individuals lived. The evolutionary impact of late-life decline therefore remained moderated by these external factors. When comparing this archaeological record to species that exhibit negligible senescence, the difference becomes clearer. In animals where adult survival is consistently high across generations, a greater proportion of individuals regularly experience advanced ages. The cumulative influence of late-life traits on reproductive success becomes more substantial. In early human populations, by contrast, the demographic proportion of elders was smaller. Selection pressures shaping maintenance systems reflected this reality. The archaeological evidence does not suggest that humans were incapable of longevity. Rather, it indicates that longevity was statistically less common. As modern medicine reduced extrinsic mortality and increased the number of individuals reaching advanced ages, age-related conditions became more visible in population statistics. The skeletal past shows that aging has always accompanied humanity, but its demographic weight has shifted. Understanding this shift helps clarify why human aging patterns differ from those of some animals whose ecological context historically supported a larger fraction of long-lived adults. When we place archaeological evidence alongside demographic modeling, a broader picture of early human aging begins to emerge. Life expectancy at birth in prehistoric populations was often low, but this measure can be misleading. High infant mortality dramatically reduced the average. Individuals who survived childhood sometimes lived into their 50s or beyond. Yet, even when accounting for this adjustment, the proportion of people reaching what we would now call advanced old age remained smaller than in contemporary societies. The population pyramid was narrow at the top. This shape carries evolutionary implications. If only a modest fraction of individuals regularly reached late adulthood, then traits affecting survival in those years exerted weaker influence on gene frequencies across generations. The biological mechanisms underlying aging did not need to be eliminated. They simply needed to permit sufficient function through the ages most likely to be lived. In skeletal assemblages from diverse regions, researchers observe variation in age distribution depending on local ecology and subsistence patterns. Agricultural communities sometimes display different demographic profiles compared to mobile hunter-gatherers. Sedentary life could increase exposure to infectious disease, while also altering nutritional patterns. Yet across many contexts, the relative scarcity of very elderly individuals persists. This pattern reinforces the idea that extended longevity, while possible, was not the statistical norm shaping long-term evolutionary calibration. By contrast, species inhabiting protected niches may maintain broader age distributions across generations. When older individuals consistently represent a substantial portion of the population, late-life traits gain demographic weight. Maintenance mechanisms that support survival beyond early adulthood influence reproductive success more directly and repeatedly. Over thousands of generations, this demographic consistency can refine aging trajectories. For humans, the transition to modern longevity occurred rapidly in evolutionary terms. Improvements in sanitation, public health, and medical intervention expanded the upper layers of the population pyramid within only a few centuries. More individuals now live into their 70s, 80s, and beyond. As a result, intrinsic aging processes have become central to societal health concerns. Conditions such as cardiovascular disease and neurodegeneration dominate mortality statistics precisely because so many people reach ages where cumulative damage becomes decisive. The archaeological record shows that the biological capacity for aging has always existed, but its demographic prominence has shifted. Comparing ancient population structures with those of species that age more slowly highlights how frequency shapes refinement. Where long life was historically common, aging trajectories could be shaped by sustained selection across many generations. Where it was less common, aging patterns remained calibrated to earlier horizons. The bones of our ancestors do not tell a story of sudden modern decline. They tell a story of changing survival probabilities. Aging in humans reflects this deep demographic history, written not only in genes but in the age structures that once defined our communities. In the span of only a few centuries, human survival patterns changed more rapidly than at almost any other point in our evolutionary history. Advances in sanitation, agriculture, medicine, and public health dramatically reduced deaths from infection, injury, and childbirth. Vaccination programs lowered childhood mortality. Antibiotics transformed once fatal bacterial diseases into treatable conditions. Improved nutrition strengthened immune defenses and supported development. As extrinsic causes of death declined, more individuals survived into middle and late adulthood. The upper layers of the population pyramid widened. With this demographic shift, intrinsic aging processes became increasingly visible. Diseases that once affected a minority of individuals who reached advanced age began to define mortality patterns across entire societies. Cardiovascular disease, certain cancers, and neurodegenerative conditions rose in prominence not because they were new, but because far more people now lived long enough for cumulative cellular damage to exert its effects. The biological mechanisms underlying these conditions had long existed, shaped by evolutionary trade-offs calibrated to earlier survival expectations. Modern longevity exposed them at scale. This transition created what researchers sometimes describe as a mismatch between evolved biology and contemporary environment. The human body evolved in contexts characterized by intermittent food availability, high physical activity, and significant external mortality. Metabolic systems adapted to store energy efficiently during times of abundance to buffer against scarcity. Immune responses evolved to combat frequent infections and injuries. Yet in many modern settings, caloric intake remains consistently high, physical exertion is reduced, and sanitation minimizes infectious threats. These changes alter how aging unfolds. Chronic low-grade inflammation, metabolic dysregulation, and vascular stress accumulate over decades in ways that ancestral conditions did not commonly reveal across large populations. The comparison with animals that age more slowly underscores how demographic continuity shapes physiological refinement. Species that have long experienced extended adult survival under relatively stable ecological conditions may have had more generations during which late-life maintenance influenced reproductive outcomes. Humans, by contrast, achieved widespread longevity only recently in evolutionary time. Our maintenance systems remain robust through early and middle adulthood, yet they were not refined over countless generations to support extreme lifespan in large populations. As modern medicine continues to extend survival, society encounters the biological consequences of this historical calibration. Aging becomes a central health concern because more individuals live into ages where intrinsic processes dominate mortality risk. The contrast with animals exhibiting negligible senescence therefore reflects not only ecological and metabolic differences, but the recency of human demographic expansion into advanced age. Our bodies were shaped in environments where living beyond certain decades was possible but not statistically widespread. Modern longevity reveals both the strength and the limits of that design. As longevity increased, the profile of disease shifted in ways that illuminate the evolutionary trade-offs embedded in human biology. In early human environments, acute infections, trauma, and complications of childbirth were frequent causes of death. These threats acted quickly. They did not require decades of cumulative molecular change to become fatal. In modern societies, where such immediate dangers are often reduced, chronic conditions emerge more prominently. Cardiovascular disease develops gradually as arterial walls stiffen and lipid deposits accumulate. Type 2 diabetes reflects long-term alterations in metabolic regulation. Neurodegenerative disorders arise from progressive cellular dysfunction in the brain. These illnesses unfold over extended periods, revealing biological processes that once had limited opportunity to influence population-level survival. The mismatch concept helps clarify this transformation. Human metabolic systems evolved under conditions of intermittent caloric intake and high physical demand. The ability to store energy efficiently was advantageous when food supply fluctuated. Hormonal pathways that promoted fat storage and insulin sensitivity supported survival during famine. In environments where caloric abundance is continuous and physical activity reduced, these same pathways can contribute to chronic metabolic strain. Over decades, the accumulation of excess energy storage influences vascular health and inflammatory processes. Aging becomes intertwined with lifestyle in ways that reflect historical calibration rather than sudden biological change. Comparing this trajectory with animals that age more slowly underscores how ecological stability shapes long-term maintenance. Species that evolved in relatively constant environments with consistent adult survival may experience fewer abrupt mismatches between ancestral and contemporary conditions. Their physiological systems remain aligned with the ecological niches they occupy. For humans, rapid cultural and technological shifts have outpaced genetic adaptation. Within only a few generations, patterns of diet, movement, and exposure to pathogens transform dramatically. The aging curve we observe today reflects this rapid transition layered upon evolutionary trade-offs established over thousands of generations. Returning to early human populations provides perspective. Physical exertion was routine. Diets varied seasonally and lacked the refined sugars and saturated fats common in modern food systems. Social bonds and environmental exposure shaped immune development differently. While individuals still experienced aging, the expression of chronic metabolic disease likely differed in prevalence and timing. Modern increases in lifespan reveal long-term vulnerabilities that were once masked by higher extrinsic mortality and different lifestyle patterns. The contrast with animals exhibiting negligible senescence highlights how continuity between evolutionary past and present environment influences aging outcomes. When ecological conditions remain stable across generations, physiological calibration persists. When conditions change rapidly, underlying trade-offs become more apparent. Human aging today therefore reflects both deep evolutionary design and recent environmental transformation. The visibility of intrinsic decline in modern populations is not evidence of failure, but of survival extended into decades where cumulative biological costs become central. Modern longevity has also reshaped the social meaning of aging. In ancestral environments, individuals who reached later adulthood represented a smaller fraction of the population. Their experience and memory held value, yet demographic weight remained concentrated among younger members. Today, in many societies, older adults form a substantial and growing proportion of the community. This shift alters how intrinsic aging processes influence not only mortality statistics but economic structures, healthcare systems, and intergenerational relationships. The biological patterns shaped by deep evolutionary history now interact with social systems designed around extended lifespan. As more individuals live into their 70s and 80s, the cumulative effects of cellular repair limits, metabolic calibration, and immune trade-offs become visible at population scale. Age-related diseases occupy increasing portions of healthcare resources. Families and communities adapt to support longer periods of late adulthood. The body's historical compromise, once embedded quietly in small groups, now influences large and complex societies. This transformation does not indicate that human biology is defective. It reflects the rapid expansion of survival into ages that were historically less common. When extrinsic mortality declines across generations, intrinsic processes naturally assume greater prominence. The rise of chronic illness is therefore linked to success in reducing early life death. In contrast, species that exhibit negligible senescence often inhabit ecological contexts where adult survival has long been stable. Their social and environmental systems evolved alongside consistent longevity. The alignment between biology and ecology remains relatively intact. Humans, by comparison, experienced a swift demographic transition driven by cultural innovation rather than genetic change. The evolutionary calibration of maintenance systems did not have millennia to adjust to this new longevity profile. As a result, modern societies confront aging patterns shaped for different statistical expectations. Returning to early human communities helps clarify the contrast. Survival depended on mobility, cooperation, and resilience to acute threats. Aging unfolded within that framework, influencing individuals but not dominating demographic structure. Today, survival extends far beyond the reproductive window for many. The intrinsic curve that once influenced relatively few now shapes the experience of millions. This does not negate the adaptive value of our evolutionary trade-offs. It reveals their context. The comparison with animals that age more slowly underscores how continuity across generations shapes refinement. Where ecological and demographic stability persist, aging trajectories align with long-term expectations. Where survival expands rapidly through cultural means, underlying biological compromises become more visible. Human aging in the modern world thus reflects both evolutionary heritage and recent transformation. If we step back from cellular pathways, demographic curves, and ecological comparisons, a broader pattern comes into view. Evolution does not aim to eliminate aging. It refines organisms to survive and reproduce within particular environmental probabilities. In humans, those probabilities historically centered on successful maturation, cooperation, child-rearing, and adaptation to unpredictable landscapes. The body that emerged from this long history is resilient, intelligent, and capable of decades of productive life. Yet it also carries limits shaped by trade-offs that once made sense in contexts where extended old age was less common. Some animals, living under different ecological pressures, followed alternative trajectories. Where adult survival was stable across generations, maintenance systems influencing late-life performance exerted stronger effects on reproductive success. Over time, this contributed to flatter mortality curves and slower observable senescence. The contrast does not imply that one strategy is superior. It reflects different solutions to the same fundamental problem: how to allocate finite energy across growth, reproduction, and repair. Humans invested heavily in cognition, social complexity, and prolonged development. These investments offered profound adaptive advantages. They enabled cultural transmission, cooperation across large groups, and technological innovation, yet they also shaped how energy was distributed across tissues and across time. The gradual rise in intrinsic mortality with age reveals this allocation. It is not a failure of design, but the outcome of priorities set by ancestral conditions. When modern medicine extended lifespan dramatically within a few centuries, the historical balance became more visible. More individuals now live long enough to experience the cumulative effects of cellular trade-offs embedded in our biology. Comparing ourselves with animals that age more slowly highlights the diversity of evolutionary calibrations rather than a hierarchy of value. Each species reflects its own ecological narrative, written across thousands of generations. Returning to early human communities grounds this understanding. Survival depended on shared knowledge, adaptability, and endurance through immediate threats. Aging unfolded within that framework, shaping individuals but not defining entire populations. Today, aging occupies a central place in public health and social planning because demographic patterns have shifted so rapidly. The deeper lesson is not that humans were designed to decline, nor that other animals escape time. It is that aging patterns emerge from historical probabilities about which ages were most often lived and which ages most strongly influenced reproduction. If some animals do not age like humans, the difference begins with how long their ancestors were expected to endure and what their survival demanded. As we reflect on the long arc of human aging, it becomes clear that what we call decline is inseparable from what we call achievement. The same evolutionary processes that limited indefinite repair also enabled extraordinary cognitive expansion and social depth. Extended childhood allowed learning to accumulate across generations. Cooperative networks permitted survival in harsh climates. The ability to plan, remember, and imagine shaped cultures that transformed landscapes. These traits required energy, time, and biological prioritization. The gradual vulnerabilities that appear in later life are intertwined with the very capacities that defined human success. In animals that age more slowly, different balances were struck. A turtle protected by its shell does not need language or symbolic reasoning to endure. A whale navigating vast oceans relies on communication and social bonds, yet its ecological niche and metabolic structure differ profoundly from those of terrestrial primates. The slower rise in intrinsic mortality seen in some of these species reflects their own allocation of resources and environmental histories. Their longevity patterns are not lessons in perfection, they are examples of alternative trade-offs. For humans, the expansion of lifespan in recent centuries has prompted a re-evaluation of what aging means. As more individuals reach advanced decades, societies confront questions of healthcare, intergenerational responsibility, and quality of life. Yet beneath these contemporary concerns lies an evolutionary story stretching back tens of thousands of years. The human body was shaped for resilience within specific statistical horizons. It was not optimized for infinite preservation, but for sufficient durability to nurture offspring, share knowledge, and adapt to shifting environments. The visibility of aging in modern populations reflects success in overcoming many external threats rather than a sudden biological decline. When comparing ourselves to animals that show negligible senescence, the contrast invites humility rather than envy. No species escapes the constraints of energy and entropy. Each follows a path carved by ecological probability and reproductive strategy. Human aging reveals the imprint of choices made unconsciously across generations. Encoded in physiology, it reminds us that survival beyond certain ages was once uncertain, and that the refinement of maintenance systems followed those probabilities. As modern conditions extend life further, the question is not whether we can erase aging entirely, but how we understand its origins and implications. If our bodies reflect a compromise between intelligence, cooperation, and repair, then aging becomes part of that narrative rather than an external flaw. The comparison with other species does not diminish humanity's path. It clarifies that longevity and decline are woven into the same evolutionary fabric that shaped who we are. When we look across the living world, the diversity of aging patterns can seem almost philosophical. Some bodies decline visibly with time, their mortality curves rising in familiar arcs. Others maintain steadier trajectories, their vulnerability increasing only gradually. Yet beneath these differences lies a shared principle. Every organism exists within limits, shaped by energy, environment, and reproduction. The variation we observe does not reflect intention or preference, but probability repeated across generations. Humans inherited a body calibrated to endure long enough to learn, to cooperate, and to raise the next generation in landscapes that were rarely stable. That calibration produced resilience through early and middle adulthood, coupled with gradual vulnerability in later decades. Other species, shaped by different hazards and rhythms, inherited alternative calibrations. Their flatter curves reveal maintenance systems tuned to ecological conditions where adult survival was reliably extended. The contrast invites reflection rather than comparison. If human aging appears more pronounced, it is partly because modern conditions allow more individuals to experience the later chapters of life. The intrinsic processes that once affected a minority now influence entire populations. Medicine, sanitation, and cultural organization have expanded the time horizon beyond what was historically common. The biological trade-offs embedded in our tissues have not changed as quickly. They remain evidence of the environments in which our species first matured. Returning to the earliest human communities offers perspective. Survival demanded mobility, shared memory, and adaptability. Aging unfolded quietly within that framework, often overshadowed by immediate risks. Today, with many external threats reduced, aging stands at the center of public attention. It shapes policy, healthcare, and personal reflection. Yet its roots remain evolutionary, tied to probabilities about which ages mattered most for gene transmission. If some animals do not age like humans, the difference begins with how their ancestors lived and how long they were expected to survive. The human body was never indifferent to time. It was simply tuned to a different set of expectations. As we consider our place among other species, perhaps the deeper question is not how to escape aging, but how to understand the balance that shaped it. If longevity and decline arise from the same evolutionary compromises that enabled consciousness and culture, what does it mean to value the years we are given within those limits?